Cell structure. The cell is covered by a cell membrane or the plasma membrane that is highly permeable and metabolically active. It is called as the basic membrane as it is trilaminar and consists of lipids, proteins and carbohydrates. The lipids. The phospholipids are made up of the head and two tails. In the head, it contains the phosphorus part. It is the polar end which is soluble in water and therefore they are hydrophilic. Therefore they are present on the outside. Whereas the two tails have non-polar ends which are insoluble in water. Therefore they are hydrophobic and they are present inside. Therefore, this forms the basis of the fluid mosaic model of the membrane. The proteins. The proteins are present either on the inner or the outer membrane. The transmembrane proteins are the proteins which are embedded inside the membrane and they project out both sides. Functions of proteins. They maintain the structural integrity of the cell. They help in transportation across membranes, therefore they act as pumps. They form passive channels through which substances diffuse through the membrane. They act as receptors for specific hormones or neurotransmitters and they also act as enzymes. So this is the outer membrane protein, inner membrane protein and the transmembrane protein which projects out on both sides. Carbohydrates. The carbohydrates attach to the proteins to form glycoproteins. The carbohydrates attach to lipids to form glycolipids. Glycocalyx is the cell coat of the carbohydrate which is present on the external surface of the cell membrane. Functions of the cell membrane. It maintains the shape of the cell. It allows movement of substances in and out of the cell. It forms a sensory surface. Outside it is positively charged and inside is negatively charged, which helps in the passage of sodium potassium leads to depolarization. Bears receptors for certain enzymes. The cell membrane of rods and cones in the retina are sensitive to light, also helps in transportation. Endocytosis is movement of molecules inside the cell. Exocytosis is movement of molecules outside the cell. Pinocytosis is movement of the fluid inside the cell. Phagocytosis is engulfing foreign material. Contacts between adjoining cells. Unspecialized contacts. Under this we have CAMs, which is cell adhesion molecules. These are glycoprotein molecules that occupy the cell membrane and are attached to the intermediate or the link proteins. Types of specialized junctions. The first one is anchoring junctions. They bind the neighboring cells together. Under this we have adhesive spots, adhesive belts and adhesive strips. Adhesive spots. These are called as desmosomes or macular adherents. They provide strong anchorage between the cells of the epidermis. The CAMs are integrins. The link proteins are desmoplakins. Adhesive belts. These are called as zonula adherents. The junction is in the form of continuous bands. No filaments are present in the gap of plasma membranes. The CAMs are cadherins. Adhesive strips are called as fascia adherents. They are in the form of short strips. They are found in smooth muscles and intercalated disc of the cardiac muscle. Hemidesmosomes. The thickening of the cell membrane is seen only on one side and these are common in basal epidermal cells. Focal spots, also called as focal adhesion plugs or focal contacts. They send signals to the cell and initiate cytoskeletal formation. The CAMs in focal spots are integrins. The intermediate proteins are alpha-actinin, vinculin and taline. Occluding junctions. These are also called as the tight junctions. They bind the cells together and act as barriers that prevent the movement of molecules into the intercellular space. There is no intercellular gap. Leaky tight junctions are where the gaps are incomplete and the junctions allow diffusion. Gap junctions are also called as communicating junctions. The plasma membranes are not in contact, but they lie close to each other. The gap contains bead-like structures. The wall of each channel is made up of nexins, which are called as connexons. These are a type of proteins. Cell organelles. The first one is mitochondria. The mitochondria has two layers, that is the outer membrane and the inner membrane. The crista are folds of the inner membrane. The inner membrane is made up of a fluid called as the matrix. The DNA is maternal inherited, which means it is derived from the ovum. The matrix contains DNA and RNA. Erythrocytes do not contain mitochondria. The function of the mitochondria is that it helps in the synthesis of ATP and also it is involved in the Krebs cycle. Therefore, it is called as the powerhouse of the cell. The mitochondria is called so because they appear as granules or rods. So these are the ribosomes, the crista, the inner membrane and the outer membrane. Endoplasmic reticulum are folded structures in the cytoplasm. We have rough endoplasmic reticulum and smooth endoplasmic reticulum. The rough endoplasmic reticulum contains ribosomes and helps in protein synthesis. The smooth endoplasmic reticulum do not contain ribosomes and helps in lipid synthesis. Functions of the endoplasmic reticulum: it helps in synthesis of proteins, glycoproteins and lipoproteins. Ribosomes. The ribosomes are present on endoplasmic reticulum or they are freely present. If they are present singly, they are called as monosomes, and if they are present in the group, they are called as polysomes. Each ribosome consists of a protein and RNA. It can either be 40S or 60S based on the sedimentation rate. Functions of ribosomes helps in protein synthesis. Golgi apparatus. These are flat membranous sacs which are made up of the cis phase, the medial Golgi and the trans phase. The cis phase is closest to the nucleus. The medial Golgi is in between. The trans phase is closest to the cell membrane. It is called as the post office mail. It helps in processing, packaging and exporting. Lysosomes. The lysosomes destroy the unwanted material inside the cell. They contain 40 different types of enzymes. The formation of lysosomes. Primary lysosomes are when the enzymes in the vesicles that are inactive due to lack of acid medium bind with endosomes containing proteins for producing acidic medium. The fusion of these two produces secondary lysosomes. The H plus ions are pumped into the vesicle, which is the acidic medium. It activates the enzymes, therefore mature lysosomes are formed. The lysosomes helps in digesting the material. Phagolysosome is phagosomes and lysosomes. Multivesicular bodies are when lysosomes fuse with pinocytic vesicles. After digestion, the waste is thrown out by exocytosis. Some remains in the cell form the residual bodies. Peroxisomes. These are membrane-bound vesicles containing enzymes. The functions: it helps in detoxification and oxidation of long-chain fatty acids, contains enzymes which are called as catalase, and it is present in the cells of liver. Nucleus. The nucleus consists of the chromatin, nucleolus, nucleoplasm, and the nuclear membrane. Chromatin is the network of fibers. Heterochromatin are irregular dark masses of chromatin which contain closed face nuclei. Euchromatin are loose light stains which contain open face nuclei. Chromatin contains proteins called as histones. Nucleosome is the coiling of DNA around the histone complex. During cell division, the chromatin inside the nucleus becomes tightly coiled and forms rods like structures called as chromosomes. Nucleoli. The nucleus contains one or more dark rounded structures, helps in the synthesis of RNA. Nucleoplasm is the space within the nucleus. Nuclear membrane is double membrane. Perinuclear space is the space between the outer and the inner membrane. The inner membrane helps in the attachment of chromosomes. Inside the inner membrane, there is a layer containing proteins and network of filament called as the nuclear lamina. The nuclear membrane consists of the nuclear pores. Cytoskeleton. It consists of the microfilaments, which is made up of the G-actin. The function: helps in maintaining the shape of the cell, alters orientation, helps in the contraction of muscles, which is sarcomeres, and the microvilli contains bundles of actin filaments. Microtubules. They contain protein called as tubulin. Function: provides stability to the cell, provides movement for cilia, facilitates transport of organelles within the cell. Intermediate filaments. It comprises of proteins which include cytokeratin, desmin, laminin, neurofilament proteins. Functions of intermediate filaments: provides mechanical strength and links the cells together. Centrioles. These are short cylinders that lie at 90 degrees to each other. Function: helps in spindle formation during cell division configuration. Chromosomes. Human beings have 23 pairs of chromosomes. It is diploid, that is, it has 46 chromosomes and one pair of sex chromosome. The sperm and the ovum have half the number, that is 23 chromosomes, therefore it is called as haploid. Females are homogametic, which is they have two X chromosomes, and the males are heterogametic, that is they have X and Y chromosome. Each chromosome forms two parallel rod-like structures called as chromatids, which is attached at the centromere. Here, the chromatids are thinnest and this region is called as primary constriction. The centromere is not midway. This results into long arm and short arm. Metacentric is when the arms are equal in length. Submetacentric is when the arms are slightly different in length. Acrocentric is when one arm is much shorter than the other. Telocentric is when the centromere may lie at one end. The part of the distal body appears rounded from the rest of the region. This is called as the satellite body. Chromosomes contain genes. Genes are functional units of stored information that helps in the development of particular features of an individual. Each cell differs from another based on the proteins present. Chromosomes are made up of DNA. DNA forms a double helix. Each strand consists of nucleotides, that is deoxyribose, sugar, base and phosphate. The nitrogenous bases include adenine, guanine, cytosine and thymine. RNA contains ribose sugar and uracil instead of thymine. Synthesis of proteins. Proteins are made up of amino acids. Each amino acid is represented by three bases, that is the triplet code. The DNA strands separate from each other. Synthesis of mRNA takes place. The code from DNA is passed to mRNA by transcription. The part of mRNA that bears the code for one amino acid is called the codon. This molecule of mRNA separates from the DNA and moves from the nucleus to cytoplasm. The cytoplasm has tRNA which attaches to one amino acid and the other side forms anticodon. tRNA along with amino acid arrange along the mRNA and this is called translation. Proteins are made up of polypeptide chains. The flow of information from DNA to RNA proteins is called as the central dogma of molecular biology.